Okay, everybody, we're going to go ahead and go through Chapter 3, which is about microscopes. So we're going to answer some questions about how small the objects are that we're going to be looking at in lab. So the standard units of microscopy are the micrometer and the nanometer. Micro is represented by the Greek letter mu, which is, it's kind of like a U with an extra little tail attached to it. So whenever you see this, it means micrometers. And then nanometers is easier, it's just NM. So that's a easier abbreviation to remember. A micrometer is 1/millionth of a meter. So it's very tiny. It's hard to really conceptualize it. If we were in person in class, I would kind of show you how long a meter is. But even trying to imagine 1,000,000 times smaller is very challenging. And then a nanometer is 1/billionth of a meter. And this is pretty much impossible for a person to even picture or conceptualize. A nanometer is much, much smaller than a micrometer. Oftentimes things in nanometers are more like, strands of DNA and very, very tiny viruses, that kind of thing. So we've already mentioned these guys, but I wanted to mention their names again. And please remember that there will be quite a few questions on Exam 1 about these different scientists and doctors and different people who contributed to the science of microbiology. So we had Robert Hooke making microscopes in the 1600s, and then Joseph Lister's father actually built microscopes in the 1800s. And of course we already mentioned Anton von Leeuwenhoek, which was the guy who really gets the credit for inventing the first microscope, even though it wasn't technically the kind of microscopes we know of today. But definitely you should know these names. Robert Hooke, we already talked about Joseph Lister, but now we're also talking about his dad, and then of course Leeuwenhoek. So the microscopes that you're going to be using in lab are called the light microscope or the compound light microscope. They use two lenses to magnify objects which are illuminated by a light source. So there's a little small light bulb in your microscope, and that's how your microscope is able to provide the light so that you can see these small objects. Light microscopes usually have 3 to 4 objective lenses. The microscopes that we have in lab have 4 of them, and we're going to talk about all the parts of the microscope and what they do and how to operate the microscopes. We're going to do all of that in lab. That'll be our first actual real lab, is working with the microscopes. The ocular lenses are the eyepieces that you look through, and they usually magnify it 10 times bigger, which is what our microscopes in lab do. So our 2 lenses are called the ocular lenses and then the objective lenses. The resolution is the ability to distinguish objects being seen under the microscope. It's also called the resolving power. So essentially the resolution is just how good you're able to see the objects under the microscope, how in focus they are. And I already said that we're going to be using a compound light microscope. So here's a picture of the microscope that we're going to be using in our lab. There's also a picture of it in the lab manual. I'm not going to test you on the parts of the microscope. You should have already covered that in A&P 1. But just as kind of a reminder, there are the parts, and we will briefly go through these parts in lab. So there will certainly be a quick review of all the microscope parts. This is actually a picture that I took from a different textbook. I've got your textbooks next to me. Let me see if your book has something similar. Let's see. Yeah, your book has something that's somewhat similar, although it's not quite as good as this. It's on page 55 in your book. So this is essentially just showing you the different different objects and what kind of microscopes can see them. So if we start over here with the human adult, obviously you can see that with the naked eye. Nerve cells, chicken egg, frog egg, and that's kind of about where it stops, right at about 1 millimeter or so. And then we get into the micrometers. The micrometers is what you can see with our light microscopes that we're going to use in lab. So things like human egg cell, epithelial or skin cell, red blood cells, typical bacteria, the nucleus, the chloroplast, the mitochondrion. So all those things we can see with our light microscopes at varying levels of magnification. Then, when we get a lot smaller into the nanometer range, we've got the electron microscopes. So we'll talk about all kinds of different types of electron microscopes in this lecture, but you guys will not be using electron microscopes at all in class. So electron microscopes gives us the ability to see very small bacteria, viruses, ribosomes, protein, amino acids, and then we haven't quite gotten to the atomic level yet, although I don't know, part of me kind of thinks that maybe some governments or some very, very, rich companies may have microscopes that can see at the atomic level. I don't know. But the technology is definitely getting better and better to the point to where we can almost see at the atomic level. I won't really test you on any of these sizes. It's just important that you guys understand that light microscopes can be used to see things in the micrometer range, and then electron microscopes, of course, they can see those same things, but electron microscopes are used for nanometers. So I may ask you a question on the test, something like, which type of microscope would you use to see, something that's measured in micrometers, and then your answer could be light microscope or electron microscope. But then if I ask you something that's in nanometers, the only possible answer is electron microscope. So this is going to be different than what you experience in your A&P courses, because we will actually be using the oil immersion objective lens in our lab, whereas you probably did not use that fourth lens in A&P 1 and 2. You probably only used the high power lens and not the oil immersion lens. So in order to see bacteria better, you have to use the oil immersion lens, which is the highest magnification strength objective lens on the light microscope. So what we're going to do in lab is, once you have something focused under the high power lens, you have to put a drop of oil on the microscope slide, and then you slide the objective lens into the oil, and that will allow you to see the object better. It will increase the magnification and resolving power, so you'll be able to see these smaller bacteria a little bit better with the oil immersion lens. As you can imagine, it can get a little bit messy because it's basically like mineral oil or baby oil. It has that same consistency. So, you have to do more than just rinse the slide off. Often the slides that we use, we're just going to throw out when we're done. But it still gets a little messy working with that oil, so just kind of be prepared for that. There's other types of light microscopes that we will not use in our class. One is called the dark field light microscope, in which the condenser blocks the light. That's used to see other parts of the cell, like the insides of the cell. There's a picture on page 57. And actually, the picture on page 57 is kind of neat because it shows you the physics involved in bending the light and how the microscope actually works internally. We won't worry about any of that in our class, but some of you guys might be interested in looking at that in the book and finding out a little bit more about how the lenses bend the light. It's kind of neat. Another type of light microscope is the phase contrast microscope, which controls the light better in order to see smaller details. Differential interference contrast is similar to phase contrast, but it uses a prism effect to create a more three-dimensional image. And And let's see, that is in your book on page 58 if you want to look at an image. Your book's pretty cool because what it does is it takes the same organism. I believe it's a paramecium. Yeah. So it looks at the paramecium under the microscope, but it looks at it under several different types of microscopes. So you can see what it looks like under the light microscope, the dark field, the phase contrast, so on and so forth. So it's neat to be able to see those differences. Fluorescence microscope uses UV light to examine certain microbes. Another one is called a confocal microscope, which uses laser light to illuminate one plane of a specimen at a time, which produces an even better three-dimensional image. And a scanning acoustic microscope uses sound waves to view cells. So the sound waves will actually bounce off the object, and then you can see the object that way. So I would say you want to know these different types of microscopes. It's not going to be a huge part of the test, but a few of them will be on there. I would say just make some flashcards with the name of the microscope and some of the details about them, and just be able to differentiate between the different types of microscopes. So now we get to the electron microscope, which is a whole nother kind of thing, a whole different type of technology. So we've got the transmission electron microscope, which uses a beam of electrons. The electrons pass through the object, and this is used to view extremely small objects such as viruses and mitochondria. The scanning electron microscope uses a beam of electrons that's reflected back in order to view extremely small objects. So really, you can use the TEM or the SEM to look at the same object. You just get a little bit different of a view. And the picture in your book is on page 60. So that is comparing the transmission electron microscope to the scanning electron microscope. They're two different types of electron microscopes that allow you to see objects a little bit differently. So a lot of you guys have probably never seen an electron microscope, and depending where you end up working or what you end up doing for a career, you may never see one. This is what one looks like. You can see that the microscope itself is very, very tall. Electron microscopes can be 5 or 6 feet tall. And then they're attached to computers, and they're attached to this huge desk. So it's basically like this whole big desk, and then the microscope is attached to the desk, and then you use the computer to control the microscope, and then usually when you're looking at something under the microscope, it comes up on a computer screen. So this is actually even a little bit older of a picture of an electron microscope. These things are very large. They are very, very expensive. They're expensive to purchase, and they're also expensive to maintain and, keep them functioning at optimum levels. So, again, you may never get to see an electron microscope in person. They're very, very important for research in viruses, and then of course moving to higher and higher levels of magnification to look at even smaller objects. So I am going to go ahead and stop the lecture here. I can only record for 15 minutes with the software that I use, which is good because then it gives you guys the opportunity to take little breaks. So I only have a few slides left, so we'll finish everything up in the second recording. Again, just make sure you're taking good notes, just as if we're in class, live and in person, and then I can address any questions or any issues when we get back to class.